What do we inherit permanently from our parents? Do you think wealth? Do you think land? Do you think house? Do you think car or bike? No, none of this. Only thing is the DNA, which is the wealth of life. Each child carries a library. Every father and mother contributes 3 billion DNA letters, which such as A, T, G, C. Each cell in child's body carries 6 billion DNA letters, which is sometimes the scientists call this as a book of life, which is very tiny, very 4 letters arranged in countless combinations. These combinations, inside the combinations, contributes to the different things in the life. Say example, how our blood carries oxygen from lungs to the other parts of the body, how our ear shapes, how our lungs and other body organs function. One of the most significant advancements happened over the past decades in science is the human genome sequencing. From that, we can read the book, we can find where the typos are, and in some cases, we can also find out which typos can cause which type of diseases. So some of the diseases are deleterious. Say example, you know the sickle cell disease and thalassemia, which are caused by a gene called hemoglobin, which carries the oxygen from lungs to other parts of the body, which is a blood disorder, which is a devastating disorder. The second one is the cystic fibrosis, which is caused by a gene called cystic fibrosis transmembrane domain, which affects the lungs in our body. Finally, you have a muscle disorder, which is caused by a gene called dystrophin, which is called DMD, dystrophin muscle dystrophy. But on the other side, you have some beneficial mutations in our body as well. There are tiny group of population across the world. They have beneficial mutations in their library, in their book of life. They are permanently resistant to HIV. So one such gene is the CCR5. Okay, CCR5 is the gene where the HIV uses to enter into our human body. So these people, since they have the typos in the CCR5 gene, so they are permanently resistant to HIV. The second example is the gene called PCSK9. Okay, the PCSK9 is the gene which is involved in the cardiovascular disease. Okay, if there is a mutation in this gene, which there is a typo, there is a genetic variation in the gene, you will be permanently resistant to the cardiovascular disease. The third example is the ApoE protein. There is a mutation in this ApoE protein, the genetic variation or the typo. Those people are permanently resistant to Alzheimer's disease. Now the question is how to fix these errors. Say example, in sickle cell disease, there is a single typo. Instead of GAG, there is a GTG. How you reverse this typo? Or in other cases like thalassemia, there is a small deletion in the gene called hemoglobin. Okay, so you need to have a tool that can scan the 3 billion bases in the genome, which is the book of life, and then cut it precisely. Either you can remove or you can repair it. Do we have such kind of tool? Yes. The tool is called CRISPR, clustered regularly interspaced palindromic repeat, which has been identified from a bacteria. One example for that is the Staphylococcus thermophilus, which is a lactic acid bacterium, which has been commonly used for fermentation of the milk products, such as yogurt or curd, which you might have heard during your lunch today. So now, like us, bacteria also get infected with viruses. Okay, so they also have to fight off the infection. How it does the infection, how fighting the infection? So it uses a system called CRISPR, the clustered regularly interspaced palindromic repeats, which is an adaptive immune system. Okay, when the virus infects the bacteria, it takes a small piece of the viral DNA, incorporates into the CRISPR array, and then when next time the same virus infects the bacteria, it uses the memory of infection, Bacteria also very smart like us. It uses the memory of infection and uses the CRISPR array system, and then it chops the viral DNA, and then virus get degraded. So couple of years back, the scientists have repurposed this CRISPR system to cure many diseases. Okay, so this is the simple CRISPR system, how it works. It is a two-component system. You have a molecular system, molecular scissors, which is called as the Cas9 enzyme, which makes the targeted double-stranded break, and then the guide RNA. The guide RNA acts like a scanner. Say, example, if you have a computer, you can just type Control F, and then you can find the search. Like that, the guide RNA searches the genome and then find out where you want to make the targeted double-stranded break. Then once you deliver these two things into the cell, it goes and finds wherever it has the binding site, and it makes the targeted double-stranded break. Okay. So what is the power of the CRISPR? So in normal healthy cells, the repair mechanism happens 1 in 1 million cells. But if you make a targeted double-stranded break using the CRISPR-Cas9, you can increase the repair efficiency by several thousand-fold. That's where the power of the CRISPR happens. Okay, so using this CRISPR, can we cure the diseases? Say, example, HIV. HIV is the most prevalent disease across the world, right? So the HIV virus, how it enters into the human cells, the T cells or the blood cells, is using a CCR5, which is a gene. Okay, it's a core receptor, right? It's like a door to enter into the human cells. If you mutate this gene using the CRISPR-Cas9, you can make it as a non-functional CCR5. It's like you are entering into this room or this auditorium using some doors. If you shut the door, you can't able to enter into this auditorium, right? So like that, CCR5 is like a door. If you mutate that CCR5 gene, if you make it non-functional, then HIV will not able to use the CCR type to enter into the cells. How can you do this? You can take the patient's body cells, you can take it into the lab, and then you can modify this using the CRISPR-Cas9, and then you can put it back into the patient. You need to have a GMP, good manufacturing practice labs. You can't do this in kitchen or garage because you need to have a very good expertise and then environment to do that. Many people think that this can be done in anywhere, but it is not at all possible. You have to do this in a specific laboratory condition. Without that, it can't be possible. The second example is the sickle cell disease, which is the most prevalent disease in this part of the world. There are around 300,000 children are born with this disease every year. Okay, so this disease, because of the single typo in the genome of 3 billion bases, normal blood cell get changed into the sickle-shaped cell. How can you reverse that? Can we use CRISPR to reverse that? Yes, it is possible. Scientists have done that. So you can use CRISPR along with the donor DNA, and then CRISPR makes the cut targeted double-stranded break, and then by homology-directed repair, you can bring back the normal function of the hemoglobin gene. It's like fixing the missing piece of the puzzle. Okay, so you can take the donor matching puzzle from another one, and then you can fix the hemoglobin gene so that you will get the complete puzzle. That is the analogy here. So the CRISPR has not only applications in biomedical science, it also has tremendous applications in agriculture and livestock. You can make the disease-resistant plants, and you can enhance the nutritional value of the plant. One example is developing an anti-browning mushroom. If you go to the US, already this has been approved in US. You can see this in the grocery store. So one of the genes which is involved in the browning of mushrooms or apple or other things is the gene called polyphenol oxidase, PPO. So what scientists have done, they have taken this tool CRISPR, and then they have mutated the polyphenol oxidase gene, and then they turned it into the anti-browning mushroom. Okay, so these are the applications. But CRISPR is not perfect. Nothing is perfect in our life, right? Nothing is ever perfect. CRISPR may produce unintended effect. When you are targeting something, there may be something else also may be happening, because you have a 3 billion bases in your cell. If you are targeting some location, there may be some other places also, there may be changes may happen. So that unintended effect may lead to deleterious events. Okay, so CRISPR is not a magic eraser, but you have to handle it properly. So it also has the ethical concerns. There are two types of genome editing. You can do it, one in somatic cells. Somatic cells is nothing but our body cells like lungs, liver, blood, or neurons. You can take the CRISPR to correct the mutation, but sometimes because of the off-target effects, this may lead to deleterious ones. But in this somatic cell editing, this may not be passed to the next generation. But if you are using in the germline or embryo editing, like say example in embryos or sperms or in reproductive cells, if you are editing, if there is an unintended effect, off-target effect, this will be passed through the next generation. So that will have the deleterious consequences. So the power of CRISPR is immense. Use it responsibly. Thank you.